Galaxy performs a variety of operations. Obviously, we cannot cover in this section over here. You might want to look into it in detail, and we have some homeworks where you will practice them by yourself and explore what Galaxy can do for you. What tasks we see here are some of them which we grab from their menus, like for example converting between coordinates. So if you have genome files, you can look into what location which genes on chromosomes they are present, and you can interplay between them. You can also do some text manipulations, along with you can convert between different formats. Here we see some of the tabs. So for example, if you click this convert genome coordinates, you can have different assemblies giving or specifying different coordinates to different genes. So if you are actually picking up a genome where you have large number of genome assemblies are available, you can correspond them with each other. Same way we can also do text manipulations, we can convert between the formats, we can filter and sort our data and other operations. For NGS tools, mainly the facilities what we can do here is quality control and manipulations. We can do some alignments and mapping, as well as we can have some BAM tools where you can have those results coming from the alignments, and you can sort those BAM files, and later on you can view them by using different views. If we convert between the formats, what facilities are here? So if we look here, we can convert tabular output into FASTA, FASTA to tabular, FASTA to FASTQ and back, and same way between BED to GFF, which are actually the assembly files, and these operations you can do here. Sometimes you have numerical data. For example, for an RNA-seq experiment, you align your reads and you quantify that expressions in terms of read counts. So you can create some graphs or histograms using this tool, filter and sort. You might also have something like where you have quality scores. You can plot them over there, or you can sort your reads based upon these numbers. You can also filter the data while specifying some thresholds on these numerical values. As far as NGS data is concerned, the main tasks which Galaxy can help with are QC, the quality control and manipulation. You can do some alignments. You can also convert or manipulate between different aligner outputs like SAM files and their BAM files, and you can visualize them. As far as QC quality control is concerned, there are different flavors of FASTQ data sets. Since we have different platforms using different quality score schemes that have different quality scores. For example, we have Sanger's scores, we have Solexa, we have Illumina. So there is a facility called as Groomer, so that can be used to convert between different FASTQ variants. You can calculate and visualize the summary statistics based upon your read qualities, and wherever you find the reads are doing bad in terms of their quality, you can trim them, or you can filter them out. As far as mapping is concerned, there are some aligners which are also available in this package. There is one which is most famous, Bowtie, that aligns Illumina and solid reads. And there is BWA, there is LASZ, MegaBlast, and there are also some short read aligners and mappers which are available. Bowtie is most famously used software. In addition to that, it has a toolkit called as NGS Picard. So that converts between different output formats and interplay between SAM and BAM files. So you can explore them also. What is SAM format? So SAM is actually the sequence alignment map. So once the aligner aligns the files, so it gives you the alignment reports in this format. So for details, you can obviously look into this web link over there. So here is a SAM format, a little bit description. So we are looking into just one read, so that was aligned by using bow tie to a reference. It's coming from Arabidopsis dataset. Here we have the read ID right here, and then we have this plus sign. So plus is where it's actually aligning to the plus strand, and the gene with which it's aligning is right here, and this is the nucleotide position at which this read aligns within that gene, and obviously the read sequence itself, and this is followed by the qualities, and then we have a number that tells us that on position number 36 there was just one mismatch. There was C in the reference, so that was replaced by G in this read. So this is how the alignment is reported. So this format is called as SAM format. Whereas the BAM format is the same alignment but in a computer readable language, so it's not man readable. So actually the alignments are reported in BAM and then they are converted into this SAM format. So there are tools that convert between these two. But in BAM format, it's easy to process them in the computers. We can have a BAM file and then we can view these alignments by using some alignment viewers. So for example, here we have an alignment viewer, a BAM view. It's v1.0.12. On the back side, we see the reference and we have some coordinates at the top, the positions in the reference where these reads are aligned. And these are our individual reads right here, here, here. So we see that the reads are on some positions, there is a cloud of reads and there are some gaps also. Maybe here we expect some insertions or deletions as far as this read data is concerned, but obviously in the reference they are insertions. So we can say we have explored indels. If it's an expression study, these all reads, if we count them together, they will give us an estimate of the expression of this underlying region. So if we compare these two regions, number 1 and 2, we can see that maybe this region is more expressed as compared to this region. There are other NGS toolbox. There is one addition to that, they call it as NGS toolbox beta. You can look into it, and there are different kind of analysis you can perform with this. So NGS data can be analyzed and explored in a lot of ways using this cool software, Galaxy.